thank you so much. Hopefully this will work. I remember being here 2 years ago. It was the first talk I had given after the pandemic, and it was a total disaster because my computer didn't work. I was so rusty. So it's nice to be back. It's nice to be here without masks, and it's of course wonderful to be in this beautiful place to talk about science. So today I'm going to tell you a story. At the beginning may be familiar to some of you, where we started just trying to make brighter dyes for a particular biological application, that's live cell single molecule tracking. But this has grown, and we're now making dyes and putting them in animals, making sensors out of these things. But at the end, I'll come back to some recently published and some new unpublished work where we've made now dyes once again for these single molecule imaging experiments in cells. All right, so I'll start with the most important slide. The goal of the lab, which is just 5 people, plus our awesome lab coordinator, Anastasia, along with open chemistry, which is a small effort, just 2 technicians right now, Carla and Sarah, where they make and provide dyes to the community. Our goal is to be a node of collaboration within Genelia and around the world. And so today I'm going to talk about projects that involve all these folks, and I'll just call it a few key names. So first, John Grimm is a senior scientist in the lab. He's done most of the chemistry that I'll talk about today. I'll start by telling you about work in single particle tracking in live cells, and that began with Bob Tejan and Xavier Darzak, along with James Liu. We've made sensors out of our fluorophores, and that's a long-standing collaboration with Eric Schreider's lab. I'll show some work from Adam Cohen at Harvard. And then I'll end with this new work, and we've enlisted Marcus in his lab to help us figure out what's going on in the photophysics, and Wes Legant, who may be here and will be here later, on some of these new probes for super resolution imaging. All right, so we're chemists, and our goal is to make useful molecules for studying biological systems, and we focus on small molecule fluorescent dyes. And as was mentioned, I started in this field almost 25 years ago now, working at molecular probes. And dye chemistry is a really interesting field or subdiscipline of chemistry because it's so old. Dye chemistry began in the Industrial Revolution back in the 19th century, and a byproduct of that revolution was this substance called coal tar. This is the junk left over after you pyrolyze coal to produce coal gas or to make coke for steel production. And it's this wonderful mixture of organic molecules. And for a while, chemists had fun distilling these things out and studying their properties. Many of them had interesting smells, and so they called them aromatic, and we still use that word aromatic to denote things like these delocalized 6-membered rings. But after a while, chemists thought, it'd be fun to make new molecules out of these things that you could get from coal tar. And one of the major proponents of this idea was August von Hofmann, who was president of the Royal College of Chemistry in London at the time. He's the guy that coined the term organic synthesis. And he had a student in his lab who was 18 at the time. This is a picture when he was a little bit older, named William Perkin. And together they cooked up this idea to make a molecule using stuff they could get from coal tar. And that molecule was quinine. This is the bitter flavor in tonic water. At the time, it was an important molecule. It's an effective antimalarial drug. And this was their proposed synthesis. And I know not everyone here is a chemist, but maybe if you can remember back to your organic chemistry days, you might realize that this is just never going to happen. And remember, though, this was before we have all our nice spectroscopy techniques that we use today, like NMR and mass spec. All they really had was elemental analysis to get molecular formula and some simple functional group tests. And from that perspective, this makes sense. There's some double bonds in both of these molecules. And if you look at the molecular formula, you can take 2 equivalents of this NL-toluidine, add some oxygen, and you'll get the molecular formula for quinine plus a water molecule. So they tried this in Van Hofman's lab. It failed miserably, of course, but for some reason, Perkin thought it would be a great idea to continue this type of research. Hofmann wasn't very enthusiastic, so Perkin built a lab in his parents' attic and did it at home. And this is a reaction he tried. He took aniline. It wasn't quite clean. It was contaminated with these toluidines, and he oxidized it. And as he's cleaning up, he notices that the solution turns this beautiful purple color, and he had accidentally made the first synthetic colored compound, which he would later call movine. And this was important. He had inadvertently discovered the first synthetic colored compound, and the fact that it was purple was a big deal. At the time, all colored compounds came from natural sources, and the best purple dye was this brominated indigo derivative, Tyrian purple, which is a natural product from these spiny sea snails. You have to harvest a bunch of these snails to get a little bit of this dye, and so purple had been always associated with riches and royalty. And so suddenly Perkin could make a very good purple dye out of cheap starting materials. And so, being smart, he promptly dropped out of graduate school, filed a patent, and started a company. And that was the beginning of modern chemical industry. And many of the fluorophores that we all use today were discovered in the subsequent decades, including these classic dyes that my lab works on, fluoresceins and rhodamines. So fluorescein was first synthesized over 150 years ago now by Adolf von Bayer's lab. And this dye has really played an important role in the development of imaging technologies. So fluorescein diacetate, what's really the first indicator used in live cells. You can load this into cells, it'll release fluorescein, and then you can use the pH sensitivity of that molecule to look at changes in the cellular environment. And then, of course, fluorescein isothiocyanate, or FITC, was an early visible, excited antibody label that really sparked the field of immunofluorescence. And then the rhodamines pop up in the patent literature a few years later, synthesized by Marie Ceresol, who was working at BASF. And again, some of the dyes that we still use today, like tetramethylrhodamine, rhodamine 110, and rhodamine B, were all described in this early patent. And so I often get questions about this. You know, if these dyes have been known for a long time, why are you still working on them? Hasn't all the chemistry been done? And the age of these dyes is something of a double-edged sword. On the one hand, the fact that we're still using these molecules in modern biological experiments is a real testament to their properties. We've had over a century to find better compounds, and we simply haven't. But from an organic chemistry standpoint, the age of these dyes is something of a problem. It means that the methods we use to build these molecules are often rooted in the 19th century. And so when I started my lab at Genelia, one thing we wanted to do is bring modern chemistry to bear on these classic fluorophores, with the idea if we could make them efficiently, we could maybe give them away. And if we could come up with gentler chemistry, maybe we could make improved derivatives with superior properties, ultimately enabling new biological experiments. And so over the years, we've come up with a variety of different ways to make fluoresceins and rhodamines. So we've used reduced or leucorhodamines as synthetic intermediates to make dyes for super resolution microscopy, like these photoactivatable things. We've used palladium catalyzed cross-coupling to turn fluoresceins into rhodamines. We've used modern renditions of old reactions to make new fluorescein derivatives, and then we can turn them into rhodamines using this approach. And we've come up with completely new ways to make rhodamine dyes by taking these dibromides, which we can synthesize in a few steps, and just clicking them onto acids or anhydrides. And so with all this chemistry in hand, we started looking for different applications. And one presented itself, and it was really almost began as a throwaway collaboration. It involved single molecule imaging in vitro. So Bob Tejan, we call him Tej, was president of HHMI and had a small lab at Genelia. And he came into my office one day and said, I want to do this experiment, look at RNA polymerase interacting with DNA affixed to a cover slip. Teej's lab had figured out a really slick way of reading out the transcript with a green dye, and so he thought, let's try and tag the RNAP with a red dye. So this is in vitro, single molecule imaging, just turf, no cells, a fairly simple system. And the idea to label the RNAP was to use the halo tag system, which then was fairly new. It's an enzyme with half the catalytic machinery taken out. It reacts specifically and irreversibly with a ligand that you can attach to a small molecule fluorophore. So you just express your protein of interest as a fusion with this tag, mix it with this ligand, and then you get a fluorescent bioconjugate where the fluorophore position and stoichiometry is well defined. And so Teej had a very simple request. He said, can you just make Sci5 halo tag ligand? This was a while back. You couldn't buy good far red fluorophores with this halo tag ligand piece attached. And this was fairly simple. We did this in a couple hours and published this nice little paper with Teej. But this piqued our interest, and we started talking to Teej and Xavier and many others who were doing single molecule imaging, not just in vitro, but also in live cells. Again, using this halo tag system to attach bright synthetic fluorophores and then watch them move around. And so this was, again, 10, 15 years ago, a fairly new idea. And it was also a time when many new microscopes were being developed that could enable live cell, single molecule imaging in 3 dimensions. And this is one example from Mats Gustafsson, this multi-focus microscope, which images in 9 focal planes simultaneously. And there have been a lot of advancements on this idea. This paper is now a decade old. But at the time, the only dye with the requisite brightness, photostability, and cell permeability for these sorts of experiments was this classic fluorophore that I mentioned earlier, tetramethylrhodamine TMR, which was in that original rhodamine patent from 1887. So this dye is pretty good. It gets into cells. It reacts with this halo tag rapidly. Its main problem is a low quantum yield, only 41%, and modest photostability. So you might think, why not use a more modern fluorophore like one of the Alexafluor dyes? And the Alexafluor dyes are great, but they weren't built for live cell applications. They were built to be antibody labels. And so they have these sulfonate groups, which in many cases can preclude entry into cells. It makes them great if you just want to label an antibody in vitro, but it makes it difficult to work in living systems. And so we set out to build a better version of this classic fluorophore that would retain its small size and cell permeability, but have the brightness and photostability of some of these more modern fluorophores. And so we thought about the photophysics. So here's tetramethylrhodamine. Of course, you can get absorption to the excited state. This then can relax back down to the ground state through emission of another photon. That's fluorescence, but that only happens 41% of the time. What about the other 59% of excited states? And so one process that competes with fluorescence is twisted internal charge transfer, or TICT, where you get electron transfer from one of these nitrogen atoms into the xanthine system, and then a twist around this carbon-nitrogen bond. This species then relaxes back down to the ground state without emitting a photon, so it competes with fluorescence. Another thing that can happen maybe 1 or 2% of the time is intersystem crossing to the triplet. This triplet then can relax back down to the ground state through sensitization of singlet oxygen. And then this singlet oxygen can do bad things to the fluorophore in this case. It can oxidize the aniline nitrogen to a radical cation, and this ultimately leads to dealkylation of the fluorophore. And this seems to be the first step in irreversible photobleaching, since dealkylated rhodamines typically show lower photostability. So we wanted to stop this process to improve brightness, this process to improve photostability. And what we realized is both of them involve pulling off an electron from this nitrogen. And so we thought if we could just make that harder, maybe we could make a better dye. And so our hypothesis was if we install azetidine instead of these dimethylamino groups, maybe we could make a better fluorophore. And so we had the tools to test this hypothesis. So we could use our chemistry to take fluorescein and turn it into this azetidine-containing rhodamine dye. And true to our prediction, we see a marked improvement in quantum yield, now 90%. And the nice thing is we're just adding a couple carbon atoms to this fluorophore, and so we don't change the spectral properties. Compared to tetramethylrhodamine, there's just a shift of 1 nanometer. But it is brighter and more photostable in live cell experiments. So under equivalent imaging conditions here in single particle tracking experiments, this dye, which we called Geneliofluor549 or JF549, is twice as bright and lasts twice as long. So you can squeeze out 4 times the number of photons just by adding a couple carbons. And this has enabled some really beautiful microscopy. This is one of my favorite examples from my colleague James Liu. So the magenta little fireflies whizzing around are more or less wild type Huntington protein with the normal number of glutamine repeats labeled with JF549 using the halo tag system. And you can see how they interact with now mutant Huntington protein that has expanded glutamine repeats and is labeled with GFP. We actually started a company using this type of imaging as a primary screen for drug discovery. All right, just as an aside, we've continued to think about other strategies to improve rhodamine dyes using this photophysical scheme. And so one idea we had was to slow down this process, plucking off this hydrogen atom. We thought, what if we installed deuterium into these N-alkyl groups? We could make this harder based on isotope effects. When we started digging into the literature, we found actually that there's a pronounced secondary isotope effect on the oxidation of amines when you have deuterium here. And so we thought maybe deuterium could improve both brightness and photostability. And this is all published, so I'll just show one example. These are the JFX dyes for extra photostability, and in some cases, these 5-membered pyrrolidine rings are better than the 4-membered azetidine rings. And this is one example. This is JFX 650, and this is just imaging in yeast, and you can see that this dye is the last man standing. All right, so getting back to these azetidine dyes, we've since figured out how to tune the spectral properties of these fluorophores. So here's this JF549, the rhodamine. If we have now the carbon analog, we get JF608. And then the silicon analog, our version of Kai Johnson's SIR dye, is JF646. And we've since found if you put stuff up here on the azetidine, we can shift things typically to the blue by as much as 25 nanometers. So if you have difluoroazetidine. And it was known that if you install fluorines on this pendant phenyl ring, you can shift things up by 20 to 25 nanometers. And we can mix and match these strategies to get really any wavelength in between. And these are general, so we can do that on the carborhodamines or silicon rhodamines. I'm not going to show you every dye we've ever made. I'll just tell you the brackets. We can go down to around 480 excitation with these azorhodamines and fluorines up here. And we can go out to 720 excitation with these phosphine oxide dyes and fluorines down here. So we made a bunch of these fluorophores. We were giving them away. We were testing them in a variety of different experiments. And what we noticed is that a sum of these fluorophores, not all, were highly cell permeable. In some cases, they would work even in animals. And other dyes, a separate subset, were fluorogenic, meaning that we would make ligands out of these. They would be quiescent, sometimes colorless. But when they found their biomolecular target, they would turn on. And we wanted to understand what was going on. If we can understand it, maybe we could make more dyes or make these even better. And this, again, is mostly published now. And what we discovered is a particular kind of nerdy property of these dyes is responsible for their performance. And that property is this equilibrium between a lipophilic lactone and a fluorescent zwitterion. So classic rhodamines that have this carboxyl group here can exist in these two forms. So here's how we typically draw rhodamine dyes with this extended conjugation. These are fluorescent, visible absorbing. But this carboxyl group can bite into the central carbon. That interrupts the extended conjugation, giving a molecule then that is colorless, non-fluorescent, not charged, and lipophilic. And so based on just our results largely in cell culture, we came up with a general rubric relating this equilibrium constant, which we defined as KLZ, to the performance of the dyes. So of course you can have dyes that are fully shifted this way. They're always fluorescent. They're nice, environmentally insensitive fluorophores. You can also have dyes that are fully in this lactone form. These are always non-fluorescent. They're largely useless for biological imaging. But in between is where the interesting stuff happens. So you can have dyes that are mostly in this zwitterionic form, but have some propensity to close up. And these often show improved permeability. They can slip through lipid bilayers and then come out the other side. That's our model. You can also have dyes that are mostly in this lactone form, but have some propensity to open up. And these are often fluorogenic. They exist in this quiescent state, but when they bind their biomolecular target, that change in environment shifts this equilibrium to the fluorescent form, so you get a high turn on. Now, the issue here is when you start looking at these core dyes with these different substituents here, there's an obvious trend going from short wavelength dyes to long wavelength dyes. You go from high KLZ values to low KLZ values. And that makes sense because the electron withdrawing character of these substituents is going to affect both the spectral properties of the fluorophore as well as this equilibrium. And what it means from a design standpoint is we want to treat the long wavelength dyes differently than the short wavelength dyes. For the long wavelength dyes, we're going to want to shift this equilibrium constant up to take, say, a fluorogenic molecule and then make something that will work in an animal. And for the short wavelength dyes, we want to shift this equilibrium down, where we can take a dye that's always fluorescent and make something that will work better in cells, or even something that's fluorogenic. And so fortunately, we can tune this equilibrium through chemical substitution. In some cases, we're also changing the color. In some cases, we are not. So for the rhodamine dyes, we start with JF549. If we install fluorines here, we get JF552. This works beautifully in mice and in yeast and in other cells where JF549 doesn't show good membrane permeability and bioavailability. We can also put fluorines up here, change the color and this equilibrium constant, get JF525, which again works well in mice and fish. And then if we combine these strategies, fluorines here and here, we get JF526, which is fluorogenic. It turns on when it binds. You can also shift the equilibrium higher by installing fluorines on this pendant phenyl ring. Here's JF571. That also causes a redshift in spectra. So for the carborhodamines, we start here, JF608. It's great in terms of permeability right out of the box. You can put fluorines up here, get JF585. This is a nice fluorogenic molecule. And then a few weeks ago, we published a paper where we have now fluorines on this pendant phenyl ring. And this is an interesting far red fluorophore that's a carborhodamine, and so could complement some of the silicon rhodamines that we've made. And then for these silicon rhodamines, you actually start down here. They're fluorogenic out of the box. You can make them more fluorogenic by just putting a single fluorine on either side. And you can also install fluorines down here to get JF669, which is nicely cell-permeable and works well in mice. You can homogeneously label neurons throughout the brain just by injection of 100 nanomoles, 70 or 80 micrograms of the halotag ligand of this dye IV into an animal. All right, and so one of the major applications of these dyes recently has been in so-called chemogenetic sensors, where you have a sensor system that has a genetically encoded component and a small molecule component. And I'll just talk about 3 of these. So in one case, we've developed a voltage-sensitive sensor, Voltron, and this is related to something Michael mentioned last night. This is based on these voltage-sensitive opsins. And typically people would put fluorescent proteins down here, and when the membrane depolarizes, you get an increase in the absorption of the opsin, so an increase in FRET and a decrease in your fluorescence signal. This was work done by Adam Cohen and Mark Schnitzer. We just replaced the fluorescent protein with the halo tag bound to a Genelia fluor dye. And so that's nicely modular. You can use a bunch of different dyes. And in particular, we like JF608 because it's nicely bioavailable. It's blue shifted enough to where you still get efficient FRET, but it's red shifted enough so you can do simultaneous optogenetic stimulation. And so this is an example from Adam Cohen using Voltron with JF608, doing very high-speed imaging, tracing this back propagating action potential from the soma after optogenetic stimulation. Another example of a chemogenetic indicator is this halo camp. So this is based on the fluorescent protein derived G camps. You have a circular permuted halo tag where the dye binding site is flanked with sensor domains here, calmodulin, and some peptide that binds calmodulin in the presence of calcium. So at elevated calcium, these two domains come together. That changes the environment around the fluorophore, and if it's a fluorogenic dye, it can turn it on. So here, you can use things like this JF635, which can show a 2 order of magnitude increase in absorption and fluorescence upon binding the halo tag. So it turns on and off. And this works fairly well in cell culture, but it's been difficult to use these dyes in intact animals. And if you remember in the previous slide, you can kind of have a fluorogenic dye or you can have a bioavailable dye, but it's hard to get both. It's an either or situation. So more recently, Eric Schreider's lab has developed a next generation halo camp. They call it Walo camp. The W for this tryptophan residue that then quenches the fluorescence of the fluorophore. So the topology is a little bit different. You have this loop here with the two sensor domains and this tryptophan. Calcium binds, it changes the conformation, and then turns the dye on, dequenching due to this tryptophan. And so here you can use any fluorophore you want, including these nicely bioavailable far red fluorophores like this JF669 halo tag ligand. And this is enabling some really cool experiments doing multicolor imaging in animals here in zebrafish, doing brain-wide imaging. And Eric has pioneered 2 or even 3 color functional imaging using this system along with fluorescent protein-based indicators. All right. So many of our latest generation fluorophores have fluorines on this pendant phenyl ring. We like this because it tunes spectra. So here's that original dye, JF549. If you then install fluorines here, you get JF571. This is a minor shift. It ends up being kind of important in some cases. So for multicolor STED imaging, this little shift to the red allows more efficient depletion with the 775 nanometer STED beam. And so you can use this dye along with a far red dye in 2 color live cell STED and just use one depletion beam. Another thing this does, as I mentioned earlier, is it tunes this equilibrium. So here's JF646, and then here's JF669. These fluorines also facilitate really easy derivatization. And this is using chemistry from the 90s called masked acyl cyanides. And so you can take this unfunctionalized dye, treat it with this reagent, and then in just a couple steps, you can make a halo tag ligand or whatever you want using a fairly mild reaction sequence. So we really like this fluorination approach. So a couple weeks ago, we published a paper where we built now a comprehensive collection of fluorophores. Some of these were known, some of these we had made before, but many of the fluorinated dyes in this chart are novel. And so we're still working through what all of these fluorophores can be used for. I just want to talk about one dye, this one down here, which we call JF657. And this dye is really a live cell compatible version of a famous fluorophore, ADO647N. So here's ADO647N as the halo tag ligand. This is a compound we made a few years ago to do Drosophila imaging in fixed tissue. And the ADO dyes often have the reactive handle here at this orthocarboxyl group. So this keeps the dye open in the fluorescent form. It also is really easy to install a linker here. You don't have to have another reactive handle down here. And it also shifts wavelengths into the red, giving this nice far red absorption. The problem is it kills that negative charge here, so you're left with a cationic species. And so if you apply this now to live cells, you find that you've made an amazing mitotracker dye. So here's mitotracker, here's this dye. These cells are expressing the halo tag protein in the nucleus, but the dye never makes it there. It just goes straight to the mitochondria. So we thought our strategy of fluorination on the bottom could do a lot of the same things. It shifts wavelengths to the red, it enforces this open fluorescent form, and it also allows us to easily derivatize the dye. And in this case, this halo tag ligand goes where we want it to go. It goes to the nucleus where the halo tag protein is expressed. So we wanted to look a bit at this fluorophore, and with my colleague James Liu at Genelia, we discovered some interesting photophysics that we're still trying to wrap our heads around, and that's why we enlisted Marcus. And so this dye is a little bit brighter than our current best-in-class fluorophore JFX650 in single particle tracking experiments. But what we noticed is if you just look at the number of localizations per cell, this JFX657 just kind of stays, where JFX650 is rapidly depleted. And so what we thought was going on is that maybe there's some reversible switching between some dark state and you reach some equilibrium. So the dyes don't really bleach, they just turn off and on. We can look at this at the single molecule level. So this is a halo tag protein labeled with this JF657 on a surface. So individual molecule, it bleaches, but when you zap it with 405, it comes back. And so our hypothesis, and we're still trying to figure out if this is true, is that we've now engineered the dye with these fluorines down here, so you get light-mediated attack by water, and then you can zap it back or it will come back spontaneously. And so it's similar to the fluorescent protein DROMPA. And so we like this because if I can send you this dye and you can cage it just by adding water and a little bit of light, that's a lot easier than us trying to make a caged dye that will work well inside cells. All right, and so finally I want to talk about some unpublished work, and that involves these silicon rhodamines. So remember, here's JF646, and we could tune this equilibrium constant by putting substituents here or here. And we wanted to extend this tuning strategy to spontaneously blinking fluorophores. So almost 10 years ago now, these hydroxymethyl derivatives of silicon rhodamine were first described. And so these undergo a transient protonation deprotonation to spontaneously blink. And so of course you can do localization microscopy with these, you label your structure, you image single molecules, and you assemble an image. But I think it's now pretty well established in the field that this HM-SIR has too high a duty cycle to be useful for many biological structures, especially if you're labeling things densely. And so if you have too many molecules on at any one given time, it becomes difficult to assemble your final image. And the issue here is that the kinetics of blinking is largely hardwired into the molecular structure. You can change it a little bit by changing the pH. It's hard to do that in live cells. You can also try and put it in a lipophilic environment like a membrane. That's been done. But really what the field needs is a whole panel of these fluorophores with different blinking kinetics, so you can choose the one that best fits the structure that you're trying to image. And so we've now come up with some really efficient chemistry that allows us to make a whole palette of these spontaneously blinking fluorophores. And so dyes with fairly electron withdrawing groups here have higher on-off ratios. And then down here, lower on-off ratios. These are useful for SOFI. These are useful for single molecule localization microscopy. And so for the aficionados, Wes Legant's lab has determined the on rates or on times and off times. So the on times are, there's a slight trend as you go down this path to more electron withdrawing substituents. This is more pronounced on the off times. And so here's the different duty cycles. And so these end up working quite well. This is unpublished work from Tim Ryan's lab using this JF635B now in living cells. And so he could do 2-color live cell localization microscopy. Here, this is labeling the membrane, and then he used a photoconvertible fluorescent protein to look at these synaptic vesicles. All right, and this is just spontaneously blinking JF629, another one of our B dyes in cells. This is from Wes Legant's lab. This is just in PBS buffer. There's nothing really going on here. It's fairly straightforward to use. And so we're about to put this on bioRxiv, but if someone wants to try it out, we're happy to send you this whole panel, and then you can figure out which on-off ratio is going to be useful for your particular structure. All right, so what's next? We have also discovered some new near-infrared excited fluorophores. This is JF698, much brighter than anything out there. We've been making more deuterated fluorophores. We've become quite interested in revisiting this old structure, these duality-containing dyes. I will say, it's nice to be at Genelia because making the starting material for this dye was a $20,000 experiment. And so sometimes deuteration can be a little challenging, but hopefully we can still give these dyes away if they prove useful. We've also made cell-permeable fret quenchers for making FLIM indicators for things like cyclic AMP. So we can get far red, just single wavelength FLIM sensors. We have now bioavailable photosensitizers that can be used in animals for targeted cell ablation or for laying down DAB in different environments. We've also been playing with using dyes as linkers to drag other useful moieties into cells, things like biotin or pharmacological agents. And as many of you know, we do strive to give everything we make that's useful away for free in an effort called Open Chemistry. So you can go to dyes.genelia.org, register and request things. And then just in the last few minutes, I'll tell you another little history story. Sometimes I wonder if, dye chemistry is really the thing we should be doing, but then if you look at history, sometimes dye chemistry can lead to better things. And so one example is the development of analgesics. So back a long time ago, before Bayer was the Bayer we know today, Bayer was a dye company, and they would put out catalogs like this with feathers for hats. And I like this page because it has a few rhodamine dyes on it. You can see how these rhodamine dyes that we use today were originally staining feathers to make colorful hats. And so Bayer had come up with this blue dye, benzoazurine G, which was competing with natural indigo, which was controlled by the British. And to make this dye, they were taking phenol and then nitrating it. You would get orthonitrophenol, which they could make into this dye. And then they would also get paranitrophenol, which they didn't know what to do with, so they were just storing it in barrels in a field next to the factory. And so someone said, we got to figure out something to do with this. They ultimately found this phenacetin molecule as a pain reliever. this ended up being toxic. They figured out, oh, this is a prodrug. It's actually this, the active molecule, which here in Europe they call paracetamol. In the US we call this acetaminophen. So Tylenol is a byproduct of dye chemistry. And another example of this is early antibiotics by Gerhard Domnack, who screened through all of Bayer's dyes and found this compound Prontosil, which was really the first antibiotic, and won him the Nobel Prize in 1939. So I'm hopeful that some of the molecules that we're making will lead to better things, like discoveries that can impact human health. So with that, thank you very much for your attention, and I'm happy to answer any questions.
So thank you very much, Luke. So this presentation is open for questions.
Thank you for your amazing talk. I had a question about your Voltrons. Are they cell permeable, and is it possible that they can sense the voltage in intracellular compartments, the membranes?
Right, so the question is about this Voltron system. Yeah, so the nice thing about the Voltron system is many of the fluorophores that we've developed are bioavailable. So you can use them in animals. So in the original paper and in subsequent papers, we've shown that you can load in mice and in zebrafish and even in flies. Flies are harder, you usually have to pipette it onto the brain. But with mice, you can express the protein. And then before each imaging bout, you inject with some of the dye, and you can then image. Typically, we inject a day before we're doing an imaging experiment, and we've done that up for 8 weeks in a mouse. Yeah, so the protein part is the halo tag fused to the opsin, and then we've found variants, and others have too, where the membrane trafficking is good. So maybe your question is, we're not using like a cell impermeant fluorophore to only get the extracellular membrane. We're relying on the membrane trafficking, and that's actually the halo tag's inside the cell. And that's because we want to use dyes that will cross multiple membranes to ultimately get to the brain. You could envision having a system where you have the halo tag on the outside. Eric's done a little bit with that. Label with a cell impermeant fluorophore, and then get perfect membrane localization. But overall, we see pretty good trafficking, so we didn't have to do that.
Thank you very much for the talk. I was interested to know if you see some prospects to develop nicely photo-switching dyes at cryogenic temperature for future cryonanoscopy applications.
Yeah, so we've thought about some of the things that we've done and whether they'll work at cryogenic temperatures. It's difficult to know. I think it's just going to require screening a bunch of fluorophores to see what we can do at those low temperatures, whether it's liquid helium or liquid nitrogen. So we're thinking about this, but we haven't yet done anything. So yeah, we're always interested in new ideas, but yeah, I think it's going to be, I don't think we even know the rules yet very well, so it's hard to design something specifically. I think we have to do some experiments and just figure out what can happen at cryogenic temperatures and what can't.
Other questions? Yeah.
Thank you for a super beautiful talk. I have a couple of questions related to the use of your dyes. We work in plants and we apply clem. So one question is if your super penetrable dyes, you have tried them in plants, because we always have a lot of problems of penetrance of available dyes. And the other one is if you have any knowledge about the chemical stability of your probe, so then they can be used for correlative light and electron microscopy, because most of the times we lose the signal in the process.
Right. So to answer your first question, there have been a few users in plants. And so I think they've actually just put some stuff in Arabidopsis, like soaked it in, put it in the water. And in some cases, they were able to get good labeling with the halo tag. I'm happy to put you in touch with those folks. So, you know, we don't have a lot of information, but there have been some successful attempts. So, you know, it's probably worth thinking about. For your other question, what we have, this is again in animals, but we have, I didn't have time to talk about today, done pulse chase labeling, where we can look at protein turnover, and we're fairly confident that the dye itself is the dye conjugate, the halotag conjugate, is fairly stable over 2 weeks in the mouse brain, for example. So we can see signal for a long time. We can do pulse chase, quantify, say, protein turnover during learning and other cool things like that. So they're reasonably stable once they're attached to the halotag. They are excreted, at least in an animal, if they're not bound to the halotag.
Okay. So I also have a question. So several of these early rhodamines, they accumulated in mitochondria spontaneously. So your molecules have lost this interesting or annoying property, I mean, depending on your research subject?
Right. Yeah. So early rhodamines that are positively charged, like rhodamine 123, which originally was developed as a ballpoint pen ink and a laser dye, and you know, someone threw it at cells, and lo and behold, it stained mitochondria. Those are where you have, say, an esterified dye, or you've taken away that extra negative charge. So our dyes that are zwitterionic, we show less mitochondrial staining. In some cases with greasier fluorophores, we do see internal membrane staining, but that's more of a lipophilicity thing. It's not driven by the membrane potential of the mitochondria.